Okay, here we are again with some of our support for teachers and technicians in developing practical skills, and this is probably one of our more sophisticated enterprises. We're going to be looking about investigating DNA, and you can see we've got 5 sections here that will be leading you through a little bit more carefully in a moment. As you can read for yourself, we're going to be using micropipettes, extracting cells, extracting the DNA from those cells, amplifying it up by PCR. We're going to look at electrophoresis techniques, and then it'll become perhaps obvious how you might want to lead your students into genomic analysis further on. You'll see we have our lab laid out, and it might be something that you might adopt as a model for yourselves, but we're going to just give you a cook's tour of each of the sections. A vital part is to use micropipettes, as we might have mentioned earlier, and we'll keep emphasising that. We can see we've got other different ways of, other different mechanisms of pipetting, but the micropipette is a crucial part for this whole enterprise. So that's something that your students need to have had some practice at beforehand. Then we're into the nitty-gritties of actually obtaining the materials for extracting DNA a little bit further down. So we have some salt solutions to make up. Clearly we've got a disinfectant to make sure we've got everything nice and clean and as sterile as possible, try and removing any erroneous DNA that we have around. We're going to spin down our samples to extract our DNA pellets. Then we're going to come along to having centrifuged those samples to obtain the DNA itself. We're going to process them by a series of heating and cooling, and then we should have our DNA extracted, ready for amplification. So we have our DNA spun down, and it's now ready to amplify that very, very small sample. We're going to use a PCR machine and the process of DNA amplification, which maybe you'll be relatively familiar with the theory. So we're going to do that. It can take over 3 hours in some cases, so there's a bit of a time budget to be had here. Having done DNA, we're going to move across to electrophoresis. We have now then our DNA samples, and our DNA samples are ready to go. We're going to analyse them using electrophoresis. Electrophoresis has been around for a long while, even in my era. We're going to obtain DNA bands and patterns to look at the differences that we have, and we're just going to show you that as technology moves on and on and on and more quickly, that still using electrophoresis techniques, but a system called flash gel, whereby the whole process of electrophoresis happens almost in a flash. It takes a matter of minutes, and we have some pretty good results to be able to analyse.